Good afternoon, everybody. My name is Sue Bruiser and I'm the Executive Director of Auckland Medical Research Foundation. We're the largest independent funder of medical research in New Zealand, and we're also a charitable trust. If you're joining us from Auckland today, I hope you're all staying home and staying safe. And our thanks go out to our incredible frontline workers who are helping to deliver services and products that we need at this time. Similarly, our thoughts are with all of the people and businesses that are so significantly impacted by this latest outbreak. So it feels very, very timely to be welcoming you to the third in our series of COVID-19 research presentations. And today we're going to hear from Dr. Dan Furkert about his research into an antiviral treatment for COVID-19. Dr. Furkert works in chemical sciences in the University of Auckland, and the research he is working on leverages existing collaborations in an effort to find an effective antiviral treatment that will be essential for the long-term clinical management of COVID-19. And we all know how essential that is going to be. So without further ado, I'd like to hand over to Dr. Furkert.
Thanks very much, Sue. And I'd like to say thanks very much to the AMRF too for helping support the work that we're doing in this area at the moment. You can see that it's quite important at this time, and hopefully we can play our small part in working to fight this disease. It's really a great pleasure to be part of this webinar series. So for the general audience, what I thought I would do is show how we fit into the scheme of work that AMRF is supporting towards COVID, and hopefully give a bit of a flavour as to the kind of work we do up at the School of Chemical Sciences at the University here in Auckland, and how we can use that to push towards useful treatments for COVID-19. So here's a slide you may have seen before. I'm sure it's been dominating the TV lately, and almost any news article you read, a picture of SARS coronavirus 2, the causative agent of COVID-19. Just to make the distinction that the SARS-CoV-2 is the virus itself, and COVID is the virus that follows from that. But I'm sure no one who can see a TV or a website needs much education on that these days. It's interesting to note the COVID-2 aspect of this, in that the SARS outbreak of 2002-2003 was essentially the same organism. If you look at the genome, it's something like 96% identical to that of the current outbreak with some slight variation. And as far as we're concerned in this particular project that I'm going to talk about today, SARS-CoV-2 is 100% identical to SARS-CoV-1. So that gives us some advantage in that the work that was begun in 2002 and 3 and stopped can be used these days as we push forwards to try and find useful treatments. So what is it that I do, and what is it that our group can do towards COVID treatment? I work in chemistry research, so organic synthesis specifically, which forms part of a larger manifold of medicinal chemistry and drug discovery. And if you look across the bottom here, I've got a little bit of information about all the different areas we might find ourselves working in. So we tend to work with bioactive substances. And in many cases, these have been isolated from nature, maybe from a plant. I've got an opioid poppy here, maybe from some kind of marine organism here. And here's a highly potent marine toxin that was isolated from algae that are present in the water column in seawater. Most of our day-to-day work is indicated here by this yellow bar. And this is chemical synthesis, so working in the lab to prepare new molecules, working out ways how to do that, training people like Greg in the picture here, how to make molecules, and working with molecular modeling and other tools that we have at our disposal to try and understand how the molecules we make can be applied to biological problems and how they might be improved, or how we can understand how they work. This can then be translated forwards into biochemistry and pharmacology. So you can see straight away that we're part of a big team. So we do our part as well as we can, but we rely implicitly on the collaborations we have in these other areas to make sure that our work is relevant. And even the team itself is quite large. So I work leading a team in chemical synthesis here at the School of Chemical Sciences. This is part of a larger group that's run by Professor Margaret Brimble. And she also has a second group over in the School of Biological Sciences that's run by Dr. Paul Harris. And they predominantly do peptide science and protein science. And you'll see how these skills in the wider group become very important later on. Being part of such a large group is a big advantage. We get the chance to have in our group a lot of well-trained people in different areas, but also we have a large network. and it's really important that places like the AMRF can be part of our network. We also have to recognize support from our collaborators across the country. In particular, in this case, Professor Vernon Ward from Otago, the virologist, who is doing the virology component of our study, so that anything we do will be sent to him for analysis and further understanding. Another group that's been really important for us is the Maurice Wilkins Centre, who maybe not directly in this particular case, but for many projects that we do in the drug discovery space have been a really important supporter and a source of network contacts for us. I should point out quite clearly at this point that my expertise is in chemical synthesis. So I'm by no means an expert in virus biology and certainly not in clinical medicine. So I'll mention some of these things in passing to try and give a bit of context to what we do. But please don't take any of my information in these outside areas as quite as authoritative as the work on chemistry. So what is it we're trying to do? This slide, I just wanted to show that the ways an antivirus response can be managed. So obviously on the left, we've got our social public health measures, and we're getting pretty familiar with those. Those of us here in Auckland under lockdown level 3 will be in our houses. The rest of the country under level 2 has a little bit more freedom. But these are things that the government or your leadership can put in place to try and reduce the spread of virus from people to people. The most important keystone usually in treatment of a virus is the development of a vaccine. So that's a prophylactic measure that's put in place in advance that should prevent the spread of virus as much as possible in a population. Where we come in is in this last box, and that's the intervention of clinical medicine when people become already sick. So ideally, there would be some antiviral drugs that are available to physicians like those in the picture there, so that once people, if they have not been vaccinated or for some reason have become exposed to the virus and become sick, there are avenues to treat them. At the moment, that's becoming a big problem in COVID-19. Many of the existing drugs have some effect or some a lot lower than expected, so that once people arrive in hospital, their symptoms can be treated, but there's nothing really effective for the virus itself. Second, I've touched on vaccines, and that's such a key part of a twin response through vaccines and antiviral drugs. I'll give a little update here. As you'll see, I borrowed this slide from CNN from 2 days ago, just to update those of you who are watching as to the process of vaccine development. And this has been completely remarkable. Normally, a vaccine takes something like 4 to 20 years, and in some cases much, much longer, to be developed from original concept through to implementation in the clinic. So if you see across the top here, we've got preclinical, which involves finding or generating the idea for the virus, so the vaccine. And these phase 1, 2, 3 studies are of increasing size as the toxicity in phase 1, the efficacy in phase 2 and 3, in larger and larger patient cohorts are examined. And it's only once you get through that process that you can look at implementing into the community. And if you look in this bottom graphic here, these red dots represent vaccines that are currently under development in different labs across the world. And you can see that pipeline is advancing pretty steadily. So in terms of virus vaccines, this is an unprecedented response. And if you take the news seriously from Russia, there's even one in the implementation category here. So we're getting pretty close to a viral vaccine being available. How long it takes to translate that into an actual health measure remains to be seen. But there's definitely grounds to be cautiously optimistic that the coming months will see those become available. So how do we fit into that? Antiviral drugs. In order to give a drug in the clinic, it has to be approved for use, and that's usually through the US Federal Drug Administration, the FDA. And the barriers in order for implementation are quite large. They have to go through the same preclinical phase 1, 2, 3 screens that we just saw for vaccines. So that takes a very long time and a lot of money. So obviously the first thing to do when an antiviral drug is needed is to move to this first screen here, repurpose. Are there any drugs that have already been approved for antiviral use for any virus or maybe a related virus by the FDA? And the big advantage of that is they may well already be in the clinic. That's where they need to be. They've been approved for use. They've been manufactured. So if it can be found by screening that these drugs are in fact in some way useful against treating viruses, they're the first port of call. Definitely this avenue has been investigated thoroughly overseas and also here in New Zealand. And efforts are continuing in this direction to redirect previous antiviral drugs towards treatment of COVID. I would say that currently there are probably only 2 drugs, one of which is shown here, that appear to have any hope of being a useful COVID treatment. A second option, also leaning on the FDA approval, is to screen all drugs that are known for any use. And again, the main benefit of that is that if any effect can be found in a positive sense, these can be used straight away, even if the original indication is quite different. And where do we come in? Third option here is design. So obviously, SARS-CoV-2 is not a new virus in terms of the SARS-CoV-1 outbreak of 2002-2003. There are a lot of related viruses, and a lot of viruses operate by a similar mechanism. That means that already in the literature, there are a number of drugs, and even 2 have been approved already by the FDA, to work against viruses that use the same kind of processes as SARS-CoV-2. So if we know how they work, and there are approved drugs that can combat this using a known mechanism, this provides us a huge advantage in a starting point for designing a specific drug that's specifically designed to treat SARS-CoV-2, and ideally should be a big improvement over one of these repurposed or screened drugs in the first 2 boxes. The downside to this, obviously, is that it will take a bit longer and a lot more investment of time and money in order to accelerate something from this early stage discovery up into use in the clinic. And if we wanted to, I guess, there is a 4th option here, which is to discover new antiviral drugs, maybe looking for inspiration from nature, maybe looking for mechanism-based approaches. But these would take even longer than the design approach, given that they would be working probably against a new mechanism. So we're focusing quite heavily on this design aspect here, and that's what the current project is going to cover. So in order to combat the virus, it helps to understand a little bit about the life cycle. So as I said, I'm by no means a virus biologist, but I have a handy cartoon here that can help us understand how it works. Viruses are quite interesting. They sit on the border of alive and non-alive, and that the little virus cartoon over here contains the genetic material in terms of, in the form of RNA to be reproduced by a host cell, but it can't do this by itself. So in order to do that, the virus attaches itself to the host cell and is brought inside here and fuses with it in this endocytosis process. What happens next is that the virus can hijack the cell machinery here so that its RNA genome in the middle, this blue squiggly line, can be unpacked, copied, and then transcribed using all the cell's machinery to produce a large polypeptide chain that is then chopped up to form proteins. And these are reassembled back into the viral material and its capsid, and then released back into the host cell, sorry, around the host cell. But these processes are all key, and there's many different ways of interrupting them so that we can disrupt the life cycle. In particular, this process here, where the genetic material is translated into a large polypeptide chain, and I've got a little cartoon on the right-hand side to help illustrate how that process works. This polypeptide here is just a bunch of amino acids all joined together into a long chain. And when the genome is originally transcribed, it's an extremely long chain that contains all the elements that the virus needs in order to form copies of itself. Part of this polypeptide forms a protease enzyme here. And in this cartoon, you can see that at the green position here is the ability to chop the polypeptide chain at a defined location. So what happens is the polypeptide locates itself into the protease enzyme here, and this green element here can cleave the chain between the 2 parts. And once those dissociate from the enzyme, they can be released and repackaged by the cell into new copies of the virus. How can we interrupt that process? If we can design a small inhibitor that competes with the native polypeptide to occupy the enzyme site, then it will no longer be able to cleave this polypeptide, and these copies that the virus needs for replication won't be produced. Further than that, if we can design an inhibitor that it binds even better than the host polypeptide, then there's a chance these will selectively bind to the enzyme and take it out of circulation. And we can even look at improving that slightly by having this cartoon represented red region here. If it binds permanently to the green cleavage part of the enzyme, then that's going to take that whole enzyme out of circulation, and it's going to mean that that one can no longer go on to form a new virus particle. So if our inhibitor has a higher affinity for the enzyme than this polypeptide, and it can bind permanently, then here is a way for us to deactivate the life cycle and reduce the amount of SARS-CoV-2 particles released into the host organism. Ideally, that's done quite early in the progression of COVID-19. It turns out that a lot of complex things from a medical perspective happen afterwards, and some of those involve the body damaging itself with its immune response. So in an ideal world, soon following diagnosis, these drugs would be given, and by preventing this life cycle from happening so frequently, would reduce the amount of the viral load on the body and give your own body and its immune system a chance to clear it. So how can we start to build towards this? I would say that a couple of drugs have been designed around this general process before. So we do have something of a roadmap to work with. And that was part of the reason that this proposal was put together, was we could use this information to do what we thought would be fast and likely to have a good chance of succeeding. So a big part of the evidence that we use is the crystal structure of enzymes and proteins from the body. So if you look on this right-hand side, we have a multicolored ribbon here. And what that represents is the protease, which was this pink part of the cartoon here on the previous slide. And inside here is an inhibitor bound at the site where the cleavage occurs. Inside this protein structure of the protease is a really nicely defined binding site. So if you can design something that resembles the polypeptide, it should bind really well there. And this crystal structure, which has been obtained of the protease, gives us that design information we need to find out how we can design something to fit well with it. We can use this as a starting point now to involve computational methods to run these investigations without having to make compounds in the lab. It's worth mentioning as well that many things can be designed to bind this enzyme quite well. What we really want to do is find something that binds very, very well and ideally permanently to take that right out of circulation. So a lot of this work was done in collaboration with a really good young PhD student, Daniel Fellner, who's just started in the lab. And using his skills in computational chemistry, he's been able to push this work forward. So how can we do that? If we look at this cartoon of the polypeptide that occurs in the virus normally, we know that that binds quite well, at least, to the protease enzyme. What we need to do is design a smaller inhibitor molecule here with this red permanent binding unit that resembles this well enough to be bound in the active site. And it turns out we can actually do this a wee bit better than nature because the protease is a wee bit non-specific. It can cleave more than one thing. So if we are careful about our design, we might be able to design something that is even better at binding to that enzyme than the native substrate. And the tools we have at our disposal are amino acids here in this top box here. If you look at these for the chemically minded among you, they have a nitrogen atom at one end and an acid at the other here. And each one has a unique functional group represented by this blob. And nature uses these to make proteins and enzymes that are both structural and functional in making up our bodies and our body processes. The beauty of these is they're recycled indefinitely in the body, but once we have them, they can be strung together into long polypeptides. So a single amino acid starts to make a polypeptide in which these functional groups are displayed here. And in the case of our enzyme, they're recognized by the enzyme as part of the binding site and can help direct a substrate into the binding pocket. In order to make something that binds permanently, we also want to consider a warhead that binds covalently or forms a permanent bond with the enzyme in that critical point. So if you look at the enzyme here as being a squiggle of protein, and again, the chemically minded among you will see that that's a sulfur atom there. That's a strong nucleophile, i.e. it wants to form a bond with something that is suitably made to accept it. And here we have a couple of examples at the orange center where that sulfur atom could attack and form a permanent bond. So using our chemical knowledge, we can use elements of our structure like this to incorporate into an inhibitor so that the bond will be permanent. Obviously, it's a bit of a trade-off. We want it to be reactive enough that it happens quickly, but not so reactive that the drug might interact with other things on its way to addressing the virus in the body. As being part of a larger group, we're quite lucky in that we have a lot of skilled people. So in particular, we have Andrew and Sung down in School of Biological Sciences who've been working hard to assemble parts of our molecules based on these amino acids. And up in chemistry, Louise has been working hard to understand how best to design these warheads and how we can prepare them to be incorporated into our inhibitors. So what Dan did, taking this information into account, is to automatically enumerate more than 10,000 small inhibitors such that they had 3 or 4 amino acids in a row, followed by one of these permanently binding warhead type units at the end. So he took those from the known set of 20 or 21 amino acids, plus some unnatural ones that we're able to synthesize in the lab, plus the 4 or 5 warheads that we're considering. And he was able to take the original crystal structure that we'd spoken about earlier, remove the ligand that was there, and test each of our compounds in here to see how well they bind. So from that, we could assemble an enormous list and prioritize which of our hypothetical compounds were binding really well to the enzyme and which were likely to form a permanent bond and deactivate it. And these form our inhibitor series, which are at the right here, which are the starting points for our synthesis. We can cross-reference this with known chemistry. We can cross-reference it with known drugs so that we can rule in things that are likely to be effective, but also rule out things that might be difficult to make or lead to off-target activity that would be undesirable. There we go. Another aspect of the computational work is this intriguing property of molecular dynamics. So in the early work that Daniel had done, we treated the enzyme as a solid block, and each of our inhibitors was bound to it. But obviously in nature, enzymes aren't solid blocks, they move. So we're able to use the computational technique of molecular dynamics, in which the parameters of the enzyme and the ligand are able to allow it to flex as it would be in nature, so that you can see the interactions over a longer term of your ligand, shown here in gold down in the bottom left, with a larger enzyme structure. So the green ribbons here are nothing but a representation of the protease that we saw earlier as a rainbow colored ribbon, but this time it's animated. What that lets us do is study more carefully these interactions of our proposed ligand with the binding site that it should fit very well. And we can start to see which parts of it fit nicely, which parts are struggling, and which parts discover new ways to interact with it that we can use. If we like, we can track those mathematically, but another way that really helps us as scientists is to look at snapshots of that molecular dynamics. So here we see this blue-gray surface at the back is the binding pocket of the enzyme that we're trying to interact with. And this stick-like figure at the top here, heading right across, is that one of our design inhibitors stuck to the binding pocket. And what we're looking to see is how well that fits with the surface. Can we see, for example, ways that parts of the structure, and I have here a large 6-membered ring is interacting with the small pocket down the bottom. Are these things that are likely to stabilize it in its interaction? Are there things that are going to help it react permanently? So we can see here's the unit on the left-hand side over here. It's going to react permanently with the sulfur atom of the enzyme. Is that in the right place? We can look at parts of the structure and see how they interact specifically with parts of that protein backbone. So it gives us a great tool for understanding how inhibitors work. And once we see that in practice, then we can start to improve our model in a never-ending cycle of iterations. So in quick summary, I've led you through roughly how we've gone about building these. It's not really a forum for technical discussion, but if there are chemistry or biology questions that people had, I'm happy to answer those later on. So starting from the top, we've been able to take the crystal structure data that's available online at the moment and screen 10,000 or more potential inhibitors that we might consider as starting points for our inhibitors of, or just a design point, sorry, for antiviral drugs. Out of those, we've managed to prioritize a number of sets, which, considering known drugs and the synthesis that we can do, look to be useful stepping stones in learning about how molecules interact with the virus. And these will go on to be tested with Professor Vernon Ward down in Otago. Down in the peptide chemistry at the School of Biological Sciences, it's usually more challenging than you might expect to bond even small units together. So the team down there of Andrew and Sung have worked hard to find ways to join the small series that we need. In particular, the residue at the very left-hand end, the left-hand blob, is one that incorporates a lot more diversity than the others. And there were some challenges there in assembling the pieces that we want. So it took quite a lot of iteration and some careful thought before the team down there were able to put those together. It looks like that what we want to achieve has been done though. So the first series of 4 of the peptides with 5 different terminal residues has been prepared. And from those, it looks promising to making those into the final compounds we need. Louise in the School of Chemical Sciences has been working on the chemistry to put the warhead together. And again, being a reactive unit, we have to think about incorporating the reactive part as late as possible so that we don't have competing reactivity with different things. And there's been a few challenges in taking the methods from the literature for her to assemble what we need. And it looks like now at the bottom here that our first inhibitor set of 16 is ready to go. So the warhead has been made and transferred across for coupling to the peptide sequences that we designed earlier. So all going well, we hope that the first inhibitors can be heading down to Otago soon to the labs of Professor Ward to see if the activity is what we expect and what we can learn from that very first series. And from then on, in the future, it's just a series of testing to find out the results from our compounds, designing better ones, understanding where others were successful or less successful, and then back into the lab to make adjustments and incorporate new ones into the compounds that we want. Obviously, this is quite early stage. We're certainly at a discovery part of the project at the moment. What we're trying to do is find a benchmark for good activity against the virus in a petri dish. Once that has been achieved or going well, then there's many layers of testing that have to be undergone after that to check that they actually are active in higher mammals, for example, mice and so on. And then finding out the other properties of the molecules that are also relevant for drug development, like can it be orally available? Is it toxic in ways we don't expect? Is the drug being delivered to the target or the part of the body that we need? You might wonder how exactly this small project is going to fit into the global efforts against COVID-19. Well, it turns out it's been a really interesting process that the global scientific community has pulled together in a way I haven't seen before in sharing information quickly to help other groups who are working in similar areas. So what we hope is we can constantly be learning from other groups overseas, but not only that, that anything we find and then go on to publish can be incorporated into other groups' work who are working in similar areas, and there definitely are some there. It's possible that through our work, we could develop something that's effective enough that at least parts of it go on to become part of a drug, or even a new drug does come out of our work. But obviously that would take a number of years of rapid investigation if we were able to discover something that was that active. Some slightly more, or less direct, I guess, effects is that by having the guys in the lab here, and I've mentioned quite a few of the team members already, and ourselves as leaders of the research effort gain a lot from being involved in this process. We're building a group of researchers here in New Zealand that have the skills in biology and chemistry and medicinal chemistry to start to understand the drug design process and what's required to make a successful drug and advance it forwards towards the clinic. And hopefully that might lead in the short-term future to the possibility of New Zealand being able to produce its own antiviral drugs to make it less dependent on the whims of overseas drug companies, certainly, and give us a wee bit of independence and be able to fight problems which, in this case, are certainly on our shores as well. So just in closing, I'd like to thank all the guys who've been involved in the project so far. I think we've had a really promising start, but we're looking forward to seeing what we can discover. So I'd like to thank them, but I'd also like to thank the AMRF for their support, which has made it all possible. And those of you who've tuned in to find out a little bit about what we've been up to.
Well, thank you very much, Daniel. You have helped to make a hugely complex research project understandable and put it into terms that I guess we can all relate to. And just wanted to reiterate the work that you're doing is absolutely vital to the long-term management of this, the COVID-19. So words like warheads, we can all imagine what warheads might look like and might mean as well. So, and really great to know that this is a collaboration with Otago and working with others. You know, all of these things that are being worked on around the world at the moment are going to lead to a breakthrough and the effective health of everybody across the globe. So, yeah, loved your animated molecular dynamics. Could almost see it all working there. So I think there's a couple of questions that have come through.
Oh, okay, I've got one here. So someone's asking about what kind of computational power does it take to do this kind of modeling? So that's a great question. Quite a lot is the short answer. So it depends on what kind of answer you're trying to get. If you're taking a solid surface of a protein and trying to dock things into it in a simplistic way, that takes a certain amount. Obviously, if you want to run 10,000 compounds, you need quite a lot of computational power in order to be able to do that. So my student, Dan, has some pretty high-powered machines, one at home and one at the university, that can accelerate that work. It turns out there's been a lot of advances in hardware that I don't fully understand in the last 2 or 3 years even, that have greatly accelerated the ability of desktop computers to do this kind of thing. He does also have access to the supercomputer NESI cluster at the university here and uses that for some parts of the work. I just while you were there, you mentioned collaboration, and I'd say it's super important. I mean, we do the best we can, but we're only a tiny cog in the overall, even in New Zealand, in the overall understanding of what can be done and how best to go about it. So without the help from the guys at Otago, you know, the different branches here, the people at SBS and FMHS in Auckland, it would be really hard to make good progress here. Was there another question? I'm not sure.
Not at this stage, but look, I'm sure, Daniel, once people have had a bit of time also to reflect on what you've been talking about, people can shoot through questions to our AMRF website, through our website as well, and contact us. So if we do have questions come through, we'll be in touch. But yeah, look, on behalf of us all the very best for your research, and we'll very, very much look forward to hopefully in the not too distant future hear back from you, hear how you're going. And,
no pressure.
Yeah, no pressure at all. And we will update everybody towards the end of the year as well, how all of these projects are getting on. So thanks again.
thank you again for the support, and thanks for the chance to coming on to explain a little bit about what we're up to. So thanks very much, Sue.
All the very best. Thank you.
Okay.
Bye now.
Bye-bye.
Bye, everyone.